Okay, here we go. Ew! Man. No blood. Again. Ah! Crikey! That hurts. Still no blood. I am going to get this to work yet. Jeez! Oh.
Hey Mark, what's going on in here?
Well, I started thinking about blood and blood type, and I didn't actually do my blood type in lab, but I can't make it work.
You can't get any blood?
I can't.
I mean, we all know you're heartless. We didn't know you were bloodless.
Well, I want to know my blood type.
Well, you don't need to do this. Remember we signed up for the 23andMe, that place that took our DNA sequence and told us all our alleles and genotypes and so on. Don't you have that up on the computer? Let's go look at that.
Yeah, I've got it right there. Let's see what I got here. Look at that.
Well, there it
Sure
is.
enough.
What are you?
Turns out, not only am I type A, but because we had our DNA analyzed, I know that I'm homozygous for the A blood type allele.
AA. Well, good for you.
It does bring, I was thinking a lot about your lecture today, and you brought up this great point about dominance and why one allele would be dominant over another. And a lot of what you said made sense, but then I thought back to blood type, and it's a little more complicated because you can be blood type A, B, O, or blood type AB. So I'm not sure, why isn't A dominant over B or B dominant over A? Why isn't there dominance?
The ABO blood group is a wonderful example of not only dominance and recessive, but maybe Mendelian inheritance. Maybe let's go to the board and let's talk
check
through this.
that
We got a
out.
few minutes. Let's go.
I do. Now I don't need to take my blood.
All right, so I think we had this left over from lab a few weeks ago, but you may remember that the ABO blood group, so there's a gene,
Right?
that's this ABO blood group gene, and it exists in 3 alleles. So that's a little different than lecture. I just always mention 2 alleles, the dominant and recessive. So this is actually a 3 allele system. So we have the IA, the A allele, the IB, or the B allele, or the little I allele. Okay? Start off with the little I allele. The little I allele is a mutation. It's actually a frameshift mutation, and you get no gene product produced.
Oh, so this is like the loss of function gene you
Exactly.
mentioned in class?
Nicely stated. Loss of function.
Okay, all right.
These other 2 alleles do code for a gene product. The IA allele codes for an enzyme that will put the A sugar on the surface of the red blood cells. The B allele will code for an enzyme that puts the B sugar on the surface of those red blood cells.
Okay, okay.
Now your question was about dominance and recessiveness.
Right.
Okay.
Right.
Well, I have a couple crosses here. Let's deal with this first cross right here, where we have 2 A-type individuals. Keep in mind, we are diploid organisms, and we will have 2 alleles at that particular gene.
Okay.
And so I made them 2 heterozygotes. And what would you
right.
think of ratio after having been through lecture today?
Well, if I'm like a pea plant, there should be a 3-to-1 ratio.
3-to-1 ratio. Let's see if that pans out. So, this individual produced 2 types of alleles. The IA allele and the little I allele, and this individual the same, right? The haploid gametes here.
All right.
Mating is a random fusion of these gametes. We will have 1/4 of the children AA, 1/4 AI, 1/4 AI, and 1/4 II.
There we go.
With regards to the A and the I allele, we do see what's known as complete dominance, sort of absolute dominance. The presence of the A allele produces the enzyme that will put the A sugar on the blood coat. I does nothing. It's a loss of function. So you're right, it is a 3-to-1 ratio here. This genotype, and this genotype will all be of the A blood type. This is actually my favorite blood type. This is the O blood type. Those are the O individuals right there. They're the homozygous recessives. All right, but the ABO blood group goes one step further. Let's take a look at this second example. Here's another heterozygote by heterozygote individual. All right?
All right.
let's just walk through what the potential, after just been through lecture, your gut reaction might be, what sort of a ratio are we going to see down here in phenotypes? Heterozygote by heterozygote.
3 to 1?
3 to 1. Well, Mendel
That's
saw
what
that.
Mendel said.
Let's see what we
That's
get
one
here.
of his rules.
Let's see what we get. So, this would be AA. This would be AB. This is AB. And this is BB.
There we go. Breaks the rule.
Let's not throw Mendel out right now. He's still got something to teach us here. Interesting situation when you have both the A and the B allele. You have a situation known as codominance. And I tried to make this point in class, is that this dominant and recessive nature doesn't happen at the level of the DNA. It's at the level of the gene product of the
Okay.
DNA. And remember, when we first started this little discussion here, is that A produces an enzyme that puts the A sugar on the surface, the B produces an enzyme that puts the B. An AB individual has both those genes functioning. You get both of them there. If you would draw a picture of that red blood cell on these individuals, you would see both the A and the B sugar. In other words, one is not dominant over the other. They are codominant. They are codominant in this situation right here. So, you thought you'd have a 3 to 1 ratio. I'd actually say that 3 to 1 ratios are really described as 1 to 2 to 1 ratios. 1 to 2 to 1. That same ratio actually holds over here. Yes, we had a 3 to 1 phenotype ratio, but if I asked you the genotype ratios, what would the genotype ratios be?
We're right back to 1 to 2 to 1.
For every
Just
one
like
of these,
this.
there are 2 of these, there's 1 of these, a 1 to 2 to 1 ratio. The only difference between these two situations is an example of complete dominance between A and I, and over here, an example of codominance between A and B. A very, very interesting situation.
So the genetics, the underlying genetics are just the same. It's about what happens to the heterozygote in expression of the phenotype. Got it.
People have been trying to disrespect Mendel for over 100 years, but he always comes through. He always comes through.
Yeah. I should know better.
All right, we are late for the faculty meeting. Mark. Come
I'm
on.
sure
You need any
it's a barn
Band-Aids?
burner.
You need Band-Aids?
I didn't draw any blood.
We'll have to look into that later. That's a problem in itself.